Hello, this is Rabie Saidi from the European Bioinformatics Institute in Cambridge, UK. I will take you through this poster to give you an update on protein functional annotation in UniProt in 2020. Our mission is to provide the scientific community with comprehensive and freely accessible information about protein sequences and functional annotation. We do this by means of our database or our knowledge base, UniProt KB, which is composed of two sections, SwissProt and Trumbull. SwissProt contains manually annotated records or proteins from literature, and it contains so far more than 500,000 proteins. Trumbull contains automatically annotated records, and it is quite larger than SwissProt. It contains 180 million records, and it keeps growing exponentially despite the mechanism of redundancy reduction that we've implemented. Records that are in Trumbull should pass through an expert manual annotation process to get into SwissProt. However, this process is slow and cannot cope with the large size of Trumbull. That is why we have implemented a parallel process of automatic annotation to enrich data in Trumbull using the SwissProt section as reference data. Automatic annotation could cover 35% of Trumbull as of the current public release 2020-03, which means 35% of proteins in Trumbull could get enriched by means of this automatic annotation. It has two approaches. The semi-automatic approach and the fully automatic approach. The semi-automatic approach is called UniRule, and it's an ensemble of 5 systems from the different sites of the UniProt consortium. The fully automatic approach is based on machine learning. Our first system was called SAS, Statistical Automatic Annotation System, which will retire in the next public release 2020-04 to be replaced by ARBA. So let's get back to UniRule. In UniRule, information on protein signatures, taxonomy, and sequence are used or is used to infer the biological roles of proteins. These rules are created, tested, and maintained by experienced curators using a platform of UniRule that is called the UniRule tool. The advantage of using UniRule is the ability to create complex rules and to take into account extra knowledge from biology and chemistry. That's why it could annotate 30 million records that could not be annotated by SAS in the current public release. However, these the number of entries that are uniquely annotated by UniRule will decrease to 8 million with the introduction of ARBA in our pipeline. So what is ARBA? ARBA stands for Association Rule-Based Annotator, and it uses subsets from the SwissProt section, transform them, and generate all possible rules with reference to a certain number of quality metrics. These rules get into a mutual selection process for filtering and then aggregated to form concise prediction model. ARBA has its own cross-validation and experiment process and also offer offers convivial graphical report for the evaluation of its prediction process. Rules are then applied to Trumbull entries the same way as it is happening for UniRule. ARBA doesn't only generate more rules than SAS, but also annotate much more entries. It also can annotate a lot of entries, like almost 40 million entries that cannot be annotated by UniRule, which results in an increase of coverage from 35 to 50%, and we will witness this in the next public release 2020-04. So ARBA doesn't only outperform SAS, but also outperform all systems that are composing UniRule. So if you want to use our rules on your data, you can use Unifier. This is an open source software that we've implemented and is available on a GitLab repository. So you can build and run the tool on your own data. It offers a format that is called URML and an engine to apply rules on your data. So you can build and run, or simply use the Docker container that is in the repository. And if you have any question, you can use our mailing list for collaboration or for any sort of queries. Thank you very much.